Welcome to Cosmic Whispers, where cosmic mysteries come alive. Your journey beyond the stars begins now. In 1903, Marie Curie stood in her makeshift laboratory, mesmerized by the ethereal blue-green glow emanating from a small glass vial. The substance inside was radium, an element she had painstakingly isolated after processing literally tons of pitchblende ore. What she held in her hands would forever change humanity's understanding of matter and energy, but it would also unleash consequences she could never have imagined. Curie's discovery of radium came at the dawn of the atomic age, when the very concept of radioactivity was still foreign to most scientists. The term itself had been coined by Curie just 5 years earlier, in 1898, to describe the mysterious energy emissions from uranium. But radium was different. It glowed with an internal light, seemingly defying the laws of physics. By producing energy without any apparent fuel source, the scientific community was captivated. Here was an element that appeared to generate heat continuously, maintaining temperatures several degrees above its surroundings. Early researchers calculated that 1 gram of radium could produce approximately 100 calories of heat per hour, indefinitely. This phenomenon suggested an almost magical source of perpetual energy, leading to wild speculation about radium's potential applications. Every great journey starts with a few brave explorers. Right now, Cosmic Whispers is just beginning, and your support means everything. If you enjoy unraveling the mysteries of the universe, hit subscribe. It's free and it helps us grow this community together. During the early 1900s, radium was hailed as a miracle substance. Entrepreneurs marketed radium-infused products ranging from toothpaste to cosmetics, claiming extraordinary health benefits. The element's ability to ionize air and emit what we now know as alpha, beta, and gamma radiation was seen as invigorating rather than dangerous. Medical practitioners began experimenting with radium treatments, believing its energetic emissions could cure everything from arthritis to cancer. But perhaps the most ambitious application proposed for radium was in agriculture. Scientists and inventors of the era wondered, if radium could energize the human body, could it also energize plant growth? The logic seemed sound to contemporary minds. Plants required energy to photosynthesize, grow, and reproduce. If radium was a perpetual source of energy, perhaps it could supplement or even replace sunlight in agricultural applications. The agricultural speculation wasn't entirely baseless. Early experiments with radioactive materials had shown that small doses of radiation could indeed stimulate biological processes. Seeds exposed to weak radioactive sources sometimes germinated faster and produced more vigorous seedlings. This phenomenon, later termed radiation hormesis, suggested that low-level radiation might act as a biological stimulant rather than a toxin. Commercial interests quickly seized upon these early findings. Patent applications from the 1910s and 20s describe elaborate schemes for incorporating radium into fertilizers, soil treatments, and even greenhouse lighting systems. Inventors envisioned radium-powered agricultural installations that could grow crops year-round, independent of weather or seasonal cycles. The radium agriculture movement gained momentum when prominent scientists began endorsing the concept. Agricultural colleges conducted preliminary experiments, and several commercial ventures were established to develop radium-enhanced farming techniques. The U.S. Department of Agriculture even funded research into radioactive fertilizers, believing they might solve food security challenges for a rapidly growing population. However, the true nature of radioactivity remained poorly understood. Scientists knew that radium emitted energy, but they didn't yet comprehend the mechanism behind this emission or its biological consequences. The discovery that radioactive decay involved the transformation of atomic nuclei wouldn't come until 1911, and the full understanding of radiation's effects on living tissue would take decades to develop. As Curie continued her groundbreaking research, she unknowingly carried radium samples in her pockets and stored them in her desk drawers. The beautiful glow that had first captivated her was actually the visible manifestation of radium's assault on the surrounding air molecules. Each photon of light represented thousands of high-energy particles wreaking havoc at the atomic level. foreshadowing the complex relationship between radiation and life that would unfold over the coming decades. To understand why radium captured the imagination of early 20th century agriculturalists, we must first examine the fundamental physics underlying radioactive decay. Radium-226, the most common isotope discovered by Curie, contains 88 protons and 138 neutrons in its nucleus. This particular arrangement creates an unstable atomic configuration that inevitably leads to spontaneous disintegration. The instability arises from the delicate balance between the strong nuclear force, which holds the nucleus together, and the electromagnetic force, which causes protons to repel each other. In radium's case, the nucleus is simply too large to maintain long-term stability. The strong force, while powerful, operates only over extremely short distances. As atomic nuclei grow larger, the electromagnetic repulsion between protons begins to overcome the nuclear binding force. When a radium nucleus decays, it releases an alpha particle consisting of 2 protons and 2 neutrons, essentially a helium nucleus. This process transforms the radium atom into radon, element 86, while releasing approximately 4.87 million electron volts of energy. The alpha particle carries most of this energy as kinetic energy, racing away from the nucleus at speeds exceeding 15,000 kilometers per second. The concept of half-life provides crucial insight into radium's behavior. Radium-226 has a half-life of approximately 1,600 years, meaning that half of any given sample will decay within this time frame. This relatively short half-life, compared to uranium's 4.5 billion years, explains radium's intense radioactivity. The more frequently atoms decay, the more radiation is emitted per unit time. Early researchers were particularly intrigued by radium's alpha emissions because these particles carry substantial energy and can be easily detected. When alpha particles strike zinc sulfide screens, they produce visible light flashes, creating the scintillation counters that became essential tools in nuclear research. However, alpha particles have limited penetrating power, being stopped by a sheet of paper or the outer layer of human skin. The biological impact of alpha radiation depends critically on where the radioactive source is located relative to living tissue. External alpha sources pose minimal health risks because the particles cannot penetrate skin. However, when radium is ingested or inhaled, alpha particles can directly damage internal organs and tissues. Each alpha particle carries enough energy to break thousands of chemical bonds as it travels through biological material. The mechanism of radiation damage involves both direct and indirect effects. Direct effects occur when radiation strikes critical molecules like DNA, causing immediate chemical changes. Indirect effects result from radiation interacting with water molecules in cells, creating highly reactive free radicals. These free radicals then attack nearby cellular components, including proteins, lipids, and genetic material. During the 1920s, scientists began recognizing that radium's biological effects were far more complex than initially assumed. The C element's chemical similarity to calcium meant that ingested radium would be incorporated into bones, where it could remain for decades. The alpha particles emitted during decay would then continuously irradiate surrounding bone marrow, potentially causing anemia, immune system suppression, and eventually bone cancer. Plants respond to radiation differently than animals, but they are not immune to its effects. Low doses of radiation can stimulate plant growth through a process called radiation hormesis, where mild cellular stress triggers beneficial adaptive responses. This hormetic effect may involve enhanced enzyme activity, improved nutrient uptake, or increased resistance to environmental stresses. However, higher radiation doses quickly become detrimental to plant health. Radiation damage to plant cells manifests in several ways: disruption of photosynthesis, interference with cell division, and corruption of genetic information. The growing tips of plants, called meristems, are particularly vulnerable because they contain rapidly dividing cells with high metabolic activity. The 1920s also saw the first systematic studies of radiation's effects on seeds and seedlings. Researchers discovered that different plant species exhibited varying sensitivity to radiation exposure. Some hardy species could tolerate relatively high doses while still producing viable offspring, while others showed immediate growth abnormalities or complete reproductive failure. Agricultural researchers were especially interested in radiation's effects on crop yields and nutritional content. Early experiments suggested that mild radiation exposure might increase certain vitamins and minerals in plant tissues, while also potentially extending storage life by killing harmful bacteria and fungi. These findings fueled optimism about radium's agricultural applications, despite growing evidence of its dangers. The scientific understanding of radiation biology remained incomplete throughout the 1920s and 30s. Researchers knew that radiation could cause both beneficial and harmful effects, but they lacked the theoretical framework to predict which outcome would occur under specific conditions. The discovery of DNA's structure wouldn't come until 1953, and the molecular mechanisms of radiation damage wouldn't be fully understood until decades later. The agricultural application of radium began not in research laboratories, but in the entrepreneurial minds of inventors and businessmen who saw vast commercial potential in Curie's glowing discovery. By 1915, several companies across the United States and Europe were actively developing radium-based agricultural products, despite the limited scientific understanding of radioactivity's biological effects. The Radium Chemical Company of Pittsburgh became one of the first major commercial ventures in this field. Founded in 1913 by entrepreneur Joseph Kelly, the company initially focused on radium extraction and purification. However, Kelly quickly recognized the agricultural market's potential and began marketing radium fertilizer, a product that contained trace amounts of radium mixed with conventional plant nutrients. Kelly's fertilizer was promoted through elaborate marketing campaigns that emphasized radium's mysterious energy-producing properties. Advertisements claimed that crops treated with radium fertilizer would grow faster, produce higher yields, and resist disease more effectively than conventionally grown plants. The company's promotional materials featured testimonials from farmers who reported remarkable improvements in crop performance after using radium-enhanced products. One of the most significant early experiments occurred at the University of California's Agricultural Research Station in Davis. Professor William Crocker led a team that exposed various crop seeds to radium radiation before planting. The 1916 study examined wheat, corn, and bean varieties, measuring germination rates, growth patterns, and final yields. Initial results were promising. Seeds exposed to low-level radium radiation showed improved germination rates and slightly increased early growth compared to control groups. Crocker's team discovered that the optimal radiation dose was critical. Seeds exposed to very low levels of radium radiation often performed better than untreated controls, while higher doses proved increasingly detrimental. This dose-response relationship suggested that there might indeed be a beneficial sweet spot for radiation exposure in agriculture, lending scientific credibility to commercial claims. The French Agricultural Research Institute at Versailles conducted parallel experiments beginning in 1917. Professor Henry Molish, working with samples provided directly by Marie Curie's laboratory, exposed potato seeds to various intensities of radium radiation. His results largely confirmed the American findings. Modest radiation exposure could enhance germination and early growth, but excessive exposure caused obvious damage to developing plants. Commercial radium agriculture reached its peak during the 1920s. The Radium Emanation Company of New York developed an elaborate system for exposing seeds to radon gas, a radioactive decay product of radium. Their process involved sealing seeds in chambers filled with radon for specific time periods, then immediately planting the treated seeds. The company claimed this method provided more controlled and consistent radiation exposure than direct radium contact. European entrepreneurs were equally enthusiastic about radium agriculture. The Radium Institute of Vienna marketed radiogenic seeds, which were exposed to radium radiation in specially designed chambers. The Institute's promotional materials claimed that radiogenic seeds would produce crops with enhanced nutritional value and extended storage life. Several Austrian and German farming cooperatives purchased these treated seeds for large-scale field trials. The most ambitious radium agriculture project was undertaken by the British entrepreneur Sir William Ramsay, who had won the Nobel Prize in Chemistry in 1904 for his work on noble gases. Ramsay established the Radium Agricultural Corporation in 1921 with the goal of creating entirely radium-powered farming systems. His vision included radium-heated greenhouses, radium-sterilized soil, and continuous crop exposure to low-level radiation throughout the growing season. Ramsay's corporation constructed a prototype facility near London, featuring glass houses with radium sources embedded in the walls and ceiling. The continuous radiation was intended to stimulate plant growth while simultaneously sterilizing the air and soil of harmful pathogens. Early trials with tomatoes and lettuce showed promising results, with plants growing faster and producing higher yields than conventional greenhouse crops. However, the economic realities of radium agriculture soon became apparent. Radium was extraordinarily expensive, with prices reaching $20,000 per gram during the 1920s. Even trace amounts added significantly to production costs, making radium-enhanced products economically viable only for high-value specialty crops. The limited supply of radium also constrained the industry's growth potential. Safety concerns began emerging as researchers gained better understanding of radiation's biological effects. Several workers at radium processing facilities developed mysterious illnesses, including anemia, bone problems, and unusual cancers. The famous case of the Radium Girls, factory workers who painted watch dials with radium paint, highlighted the element's dangers and led to increased scrutiny of all radium applications. By 1925, the initial enthusiasm for radium agriculture was beginning to wane. Long-term studies suggested that while low-level radiation might provide short-term growth benefits, the effects were often temporary and sometimes followed by unexpected complications. Plants that initially thrived under radiation exposure sometimes exhibited reduced fertility or produced offspring with genetic abnormalities. The phenomenon that initially gave radium agriculture its scientific credibility is known today as radiation hormesis, a biological response where low doses of potentially harmful agents actually stimulate beneficial effects. This concept challenges the traditional assumption that all radiation exposure is inherently damaging, suggesting instead that living organisms have evolved sophisticated mechanisms to not only survive, but potentially benefit from mild radiation stress. Radiation hormesis operates through several interconnected cellular mechanisms. When plants are exposed to low levels of ionizing radiation, their cells activate stress response pathways that enhance overall cellular function. These pathways include the upregulation of antioxidant enzymes, improved DNA repair mechanisms, and enhanced metabolic efficiency. The result is often a temporary boost in growth rate, disease resistance, and reproductive success. The molecular basis of radiation hormesis begins with the interaction between ionizing radiation and cellular water. When radiation strikes water molecules within plant cells, it creates reactive oxygen species including hydroxyl radicals, hydrogen peroxide, and superoxide ions. In high concentrations, these molecules cause severe cellular damage. However, at low concentrations, they function as signaling molecules that trigger beneficial adaptive responses. Plant cells have evolved elaborate antioxidant systems to manage oxidative stress. These systems include enzymes like catalase, peroxidase, and superoxide dismutase, as well as non-enzymatic antioxidants, such as ascorbic acid, glutathione, and tocopherols. When exposed to mild radiation stress, plants often increase production of these protective compounds, resulting in enhanced overall cellular resilience. The hormetic response also involves changes in gene expression patterns. Low-dose radiation exposure can activate transcription factors that regulate stress response genes, leading to increased production of protective proteins. Some of these proteins enhance DNA repair capacity, while others improve cellular energy metabolism or strengthen cell wall structure. The net effect is often improved plant vigor and stress tolerance. Photosynthesis, the fundamental process by which plants convert light energy into chemical energy, can also be influenced by radiation hormesis. Low levels of ionizing radiation may enhance certain aspects of the photosynthetic process, possibly by improving electron transport efficiency or increasing chlorophyll production. However, this enhancement occurs only within a narrow range of radiation exposure, beyond which photosynthetic damage becomes inevitable. The timing of radiation exposure critically affects the hormetic response. Plants are most sensitive to radiation during periods of rapid cell division, such as germination and early growth. Interestingly, these are also the periods when hormetic benefits are most pronounced. Seeds exposed to low-level radiation often show improved germination rates and more vigorous early growth, possibly because the radiation exposure activates cellular repair and enhancement mechanisms just when they are most needed. Different plant species exhibit varying degrees of radiation sensitivity and hormetic response. Generally, plants with larger genomes and more complex cellular structures are more sensitive to radiation damage. Conversely, species that have evolved in naturally radioactive environments often show greater tolerance and more pronounced hormetic responses to low-level radiation exposure. The hormetic dose range is typically very narrow, often spanning just 1 to 2 orders of magnitude. For most plant species, beneficial effects occur at radiation doses between 0.1 and 10 Gray, while doses above 50 Gray almost invariably cause severe damage or death. This narrow therapeutic window explains why early radium agriculture experiments produced such variable results. Environmental factors also influence the hormetic response. Plants grown under optimal conditions of temperature, humidity, and nutrition are more likely to exhibit beneficial responses to low-level radiation. Stressed plants, on the other hand, may lack the cellular resources needed to mount effective adaptive responses, making them more susceptible to radiation damage even at low doses. The hormetic response is not limited to growth enhancement. Some studies have shown that low-level radiation exposure can improve plant resistance to other environmental stresses, including drought, salinity, and pathogen attack. This cross-protection effect suggests that radiation hormesis activates general stress response mechanisms rather than specific radiation resistance pathways. Modern research has revealed that radiation hormesis involves complex interactions between multiple cellular signaling pathways. The initial radiation exposure triggers a cascade of molecular events that can persist for days or weeks after the exposure ends. This temporal aspect explains why plants often show delayed responses to radiation treatment, with benefits becoming apparent only after several days or weeks of growth. The discovery of radiation hormesis has important implications for understanding how life has evolved in Earth's naturally radioactive environment. Background radiation from cosmic rays, naturally occurring radioactive elements, and radon gas may have played a role in shaping the evolution of cellular stress response mechanisms. Some researchers speculate that the hormetic response represents an evolutionary adaptation to the planet's inherent radioactivity. However, the existence of radiation hormesis does not vindicate the early radium agriculture movement. The extremely narrow dose range for beneficial effects, combined with radium's tendency to bioaccumulate and its long-term health risks, make it unsuitable for agricultural applications. Modern understanding of radiation biology has revealed that the apparent benefits observed in early experiments were likely outweighed by subtle long-term consequences that were not immediately apparent. The romantic vision of radium-enhanced agriculture began to crumble in the late 1920s, as mounting evidence revealed the devastating health consequences of radium exposure. The turning point came through the tragic stories of industrial workers, particularly the Radium girls, whose suffering exposed the lethal nature of what had once been hailed as a miracle element. The Radium girls were female factory workers employed by companies like the Radium Luminous Materials Corporation and the United States Radium Corporation. Their job involved painting watch dials and instrument panels with radium-based luminescent paint. To create fine brush points for detailed work, they were instructed to lick their brushes, unknowingly ingesting significant quantities of radium with each stroke. By 1925, dozens of these workers had developed mysterious and devastating illnesses. Their symptoms included severe anemia, bone fractures that refused to heal, necrosis of the jawbones, and various forms of cancer. The radium they had ingested was preferentially absorbed by their bone tissue, where it replaced calcium in the mineral matrix. Once embedded in bone, radium continued emitting alpha particles for decades, continuously irradiating surrounding tissue. The medical consequences were horrific and progressive. Radium's alpha particles destroyed bone marrow, the body's blood-producing factory, leading to severe anemia and immune system collapse. The radiation also caused osteosarcoma, an aggressive form of bone cancer that was extremely rare in young adults. Many victims suffered from radium jaw, a condition where the jawbone literally disintegrated due to continuous radiation exposure. Dr. Harrison Martland, a pathologist who studied these cases, coined the term radium poisoning and established the first scientific understanding of radium's biological effects. His research revealed that radium was approximately 100,000 times more toxic than previously believed. Even tiny amounts measured in micrograms could cause fatal illnesses decades after initial exposure. The agricultural implications of these discoveries were immediately apparent. If radium could cause such devastating effects in human workers, what consequences might radium-treated crops have for both the plants themselves and the people who consumed them? Agricultural researchers began conducting long-term studies to assess the safety of radium-enhanced farming practices. Studies of radium-treated agricultural plots revealed alarming patterns of environmental contamination. Unlike conventional fertilizers that are absorbed by plants or washed away by rain, radium persisted in soil for centuries due to its 1,600-year half-life. Continuous application of radium-containing fertilizers led to progressive contamination of farmland, creating long-term environmental hazards. The bioaccumulation of radium in food crops posed additional concerns. Plants absorbed radium from contaminated soil, concentrating it in their tissues. Leafy vegetables showed particularly high radium uptake, while root vegetables could accumulate significant amounts in their edible portions. This meant that consumers of radium-enhanced crops were potentially exposed to dangerous levels of internal radiation. Animal studies conducted during the 1930s confirmed these fears. Laboratory rats fed radium-contaminated grain developed similar symptoms to the radium girls, including anemia, bone damage, and cancer. The research demonstrated that even small amounts of radium in food could cause serious health problems over time, particularly in growing animals with high calcium turnover rates. The environmental persistence of radium created additional complications. Unlike organic pollutants that eventually break down, radioactive contamination remains hazardous for multiple generations. Farmland treated with radium fertilizers in the 1920s remained contaminated well into the 21st century, requiring expensive remediation efforts in some cases. Water contamination emerged as another serious concern. Radium applied to agricultural fields could leach into groundwater systems, contaminating drinking water supplies for entire communities. The discovery of radium in well water near former radium agriculture sites led to widespread public health investigations and costly water treatment programs. The insurance industry played a crucial role in ending radium agriculture by refusing to cover agricultural operations that used radioactive materials. Insurance companies, faced with mounting liability claims from radium-related health problems, recognized the enormous potential costs of future lawsuits and withdrew coverage from radium agriculture ventures. Legal challenges also mounted against companies promoting radium agriculture. Farmers who had purchased radium-enhanced products began filing lawsuits claiming fraud and misrepresentation. Several high-profile court cases established legal precedents, holding manufacturers liable for failing to disclose known health risks associated with their products. The regulatory response was swift and decisive. By 1935, most Western nations had banned the use of radium in consumer products and agricultural applications. The United States established strict licensing requirements for radium possession and use, effectively ending commercial radium agriculture. Similar regulations were implemented across Europe and other developed nations. The scientific community also turned against radium agriculture as research revealed the complex ways radiation could damage living systems. The discovery of genetic mutations in plants exposed to radiation raised concerns about potential impacts on food safety and agricultural sustainability. Studies showed that radiation could cause chromosomal abnormalities, affecting plant reproduction and potentially creating unstable genetic variants. The collapse of radium agriculture marked the end of an era of dangerous optimism about radioactive materials. The industry's failure served as a cautionary tale about the importance of thorough safety testing before implementing new technologies, particularly those involving poorly understood physical phenomena. The legacy of radium agriculture continues to influence modern agricultural policy and environmental regulation. The mistakes of the radium era contributed to the development of comprehensive testing requirements for new agricultural products and established the principle that environmental and human health must be carefully evaluated before widespread adoption of any new technology. While the radium agriculture experiments of the early 20th century ended in disaster, modern science has found safe and beneficial ways to harness nuclear technology for agricultural advancement. Today's applications are based on decades of research into radiation biology, precise dose control, and comprehensive safety protocols that were entirely absent from the radium era. Food irradiation represents one of the most successful modern applications of nuclear technology in agriculture. Unlike the dangerous radium fertilizers of the past, food irradiation uses controlled doses of gamma rays, electron beams, or X-rays to eliminate harmful pathogens without making food radioactive. The process works by damaging the DNA of bacteria, viruses, parasites, and insects, preventing them from reproducing and causing illness. The World Health Organization and the Food and Agriculture Organization have extensively studied food irradiation safety, concluding that foods treated with doses up to 10 kg pose no health risks and retain their nutritional value. This technology can extend shelf life, reduce food waste, and eliminate dangerous pathogens like Salmonella and E. coli without chemical preservatives. Controlled atmosphere facilities for food irradiation bear no resemblance to the crude radium chambers of the 1920s. Modern irradiation plants use precisely calibrated cobalt-60 sources or linear accelerators that can be turned on and off like switches. Multiple safety systems prevent accidental exposure, and sophisticated dosimetry ensures exact radiation doses. The entire process is monitored by nuclear regulatory agencies and international organizations. Isotope tracers have revolutionized agricultural research by allowing scientists to track the movement of nutrients through plants and soil systems. Radioactive isotopes of elements like phosphorus, nitrogen, and carbon can be incorporated into fertilizers or plant nutrients, then followed using sensitive detection equipment. This technique has led to major improvements in fertilizer efficiency and crop nutrition. Unlike the crude radium applications of the past, isotope tracer studies use extremely small amounts of carefully selected radioactive materials with short half-lives. Phosphorus-32, for example, has a half-life of just 14 days, ensuring that any radioactivity disappears quickly after experiments conclude. These tracers provide invaluable information about plant nutrient uptake, soil chemistry, and fertilizer fate without creating long-term contamination. Mutation breeding, or mutagenesis, represents another successful application of radiation in agriculture. This technique uses controlled radiation exposure to induce genetic mutations in crop plants, creating new varieties with improved characteristics. Unlike the random radium exposures of the early 1900s, modern mutation breeding uses precise radiation doses and sophisticated screening methods to identify beneficial mutations. Over 3,000 crop varieties developed through Mutation breeding are currently grown worldwide, including rice varieties that feed hundreds of millions of people. The International Atomic Energy Agency coordinates global mutation breeding programs, ensuring that safety protocols and scientific standards are maintained. These programs have created drought-resistant crops, disease-resistant varieties, and plants with improved nutritional content. Nuclear techniques have also revolutionized pest control through the sterile insect technique, or SIT. This approach involves raising large numbers of pest insects in laboratories, sterilizing the males with precisely controlled radiation doses, then releasing them into wild populations. The sterile males mate with wild females, but produce no offspring, gradually reducing pest populations without chemical pesticides. SIT programs have successfully eliminated or controlled major agricultural pests across millions of hectares. The technique has been particularly effective against fruit flies, which cause billions of dollars in crop damage annually. Unlike chemical pesticides, SIT is species-specific and environmentally friendly, causing no harm to beneficial insects or other wildlife. Modern soil and plant analysis relies heavily on nuclear analytical techniques like neutron activation, analysis, and X-ray fluorescence spectroscopy. These methods can detect trace elements in soil and plant samples at extremely low concentrations, helping farmers optimize nutrition programs and identify deficiencies before they affect crop yields. These analytical techniques are far more sensitive and accurate than any methods available during the radium agriculture era. They can detect parts per billion concentrations of essential nutrients, toxic elements, and environmental contaminants, providing information that was impossible to obtain in the early 20th century. Nuclear technology also supports modern fertilizer production through isotope ratio analysis, which helps optimize manufacturing processes and ensure product quality. This application demonstrates how nuclear science can improve conventional agriculture without direct radiation exposure to crops or consumers. The development of radiation-resistant crop varieties has emerged as an important research area, particularly for regions with high natural background radiation or potential nuclear accidents. These studies help scientists understand the fundamental mechanisms of radiation damage and recovery in plants, knowledge that could prove valuable for agriculture in extreme environments or space exploration. Modern agricultural applications of nuclear technology are strictly regulated by national and international agencies. Comprehensive safety protocols, environmental monitoring, and health surveillance ensure that today's applications avoid the mistakes of the radium era. Regular inspections, licensing requirements, and mandatory training programs maintain the highest safety standards. The contrast between modern nuclear agriculture applications and the radium experiments of the early 1900s illustrates how scientific understanding and safety culture have evolved. Today's applications are based on comprehensive knowledge of radiation biology, precise dose control, sophisticated safety systems, and decades of research into long-term effects. The rise and fall of radium agriculture serves as a powerful reminder of how scientific enthusiasm, commercial interests, and incomplete knowledge can combine to create dangerous situations. The story of Curie's discovery and its misapplication in farming offers crucial lessons for how society should approach emerging technologies, particularly those with the potential for both great benefit and great harm. The initial appeal of radium agriculture was understandable, given the scientific context of the early 20th century. Researchers had discovered an element that seemed to violate fundamental physical laws by producing energy indefinitely. The logical extension of this apparent miracle to agriculture seemed reasonable to scientists who lacked our modern understanding of atomic physics and radiation biology. However, the radium agriculture movement also exhibited classic warning signs that modern scientists and policymakers have learned to recognize. The technology was promoted primarily by commercial interests rather than independent researchers. Safety testing was minimal and short-term. The biological mechanisms underlying claimed benefits were poorly understood. Regulatory oversight was virtually non-existent. The economic pressures driving radium agriculture adoption paralleled patterns. Seen in many modern technological controversies, companies invested heavily in radium-based products and had strong financial incentives to downplay safety concerns. Marketing campaigns emphasized dramatic benefits while minimizing potential risks. Scientific uncertainty was exploited to justify continued use despite mounting evidence of danger. The tragedy of the radium girls and the collapse of radium agriculture led to fundamental changes in how society approaches technological innovation. The development of comprehensive safety testing protocols, independent regulatory agencies, and liability frameworks all trace their origins partly to lessons learned from radioactive consumer products. Modern agricultural biotechnology benefits enormously from these historical lessons. The development of genetically modified crops, for example, involves years of safety testing, environmental impact assessments, and regulatory review before commercial approval. While controversies remain, the scientific rigor applied to modern agricultural innovations far exceeds anything attempted during the radium era. The precautionary principle, which suggests that potential harm should be thoroughly investigated before new technologies are widely adopted, represents a direct response to disasters like radium agriculture. This principle acknowledges that scientific knowledge is always incomplete and that irreversible harm can result from premature adoption of poorly understood technologies. Climate change and global food security challenges are driving renewed interest in agricultural innovation, creating both opportunities and risks reminiscent of the radium era. Emerging technologies like gene editing, synthetic biology, and artificial intelligence promise revolutionary improvements in crop productivity and sustainability. However, these same technologies raise complex questions about safety, environmental impact, and social consequences. The development of CRISPR gene editing technology illustrates how modern science has learned from historical mistakes. Extensive safety research preceded agricultural applications. Regulatory frameworks were established before commercial deployment, and ongoing monitoring systems track potential unintended consequences. While debates continue, the scientific approach to gene editing reflects the cautious, evidence-based methodology that was absent from radium agriculture. Vertical farming and controlled environment agriculture represent modern attempts to revolutionize food production through technology. These systems use LED lighting, automated nutrient delivery, and sophisticated environmental controls to grow crops in urban settings or harsh climates. Unlike radium agriculture, these technologies are based on well-understood scientific principles and extensive safety testing. The integration of artificial intelligence and machine learning into agriculture offers unprecedented opportunities to optimize crop production while minimizing environmental impact. AI systems can analyze vast amounts of data from sensors, satellites, and weather stations to provide precise recommendations for irrigation, fertilization, and pest management. These technologies promise to deliver the productivity improvements that radium agriculture falsely claimed to offer. Space agriculture presents unique challenges that echo some aspects of the radium story. Growing food in the radiation environment of space requires innovative approaches to protecting plants from cosmic rays and solar radiation. However, modern space agriculture research is conducted with full awareness of radiation hazards and comprehensive safety protocols. The story of radium agriculture also highlights the importance of public engagement in technological decision-making. The early 20th century public had little voice in decisions about radium applications, leading to widespread exposure to dangerous materials. Modern democratic processes, while imperfect, provide mechanisms for public input on emerging technologies before widespread adoption. International cooperation has become essential for managing agricultural technologies with global implications. Organizations like the International Atomic Energy Agency, the World Health Organization, and the Food and Agriculture Organization coordinate research, safety standards, and regulatory approaches across national boundaries. This international framework helps prevent the kind of uncoordinated and dangerous experimentation that characterized the radium era. The radium agriculture story ultimately demonstrates both the potential and the perils of scientific innovation. Marie Curie's discovery of radium was one of the greatest scientific achievements in human history, leading to advances in medicine, energy, and our understanding of matter itself. However, the premature and poorly controlled application of this discovery to agriculture caused environmental contamination and health problems that persisted for decades. As we face current challenges in feeding a growing global population while protecting environmental sustainability, the lessons of radium agriculture remain relevant. Promising new technologies must be thoroughly tested, carefully regulated, and gradually implemented with comprehensive monitoring systems. The enthusiasm for innovation must be balanced with respect for complexity, uncertainty, and the potential for unintended consequences. The future of agriculture will undoubtedly involve technologies that seem as revolutionary to us as radium seemed to scientists in 1903. By learning from the mistakes of the radium era, while preserving the spirit of scientific inquiry that drove Curie's discoveries, we can work toward agricultural innovations that fulfill their promise, while avoiding the tragic consequences of premature adoption.